Good morning, good afternoon, and good evening, everyone, and welcome to the Scientist.com and Biointron webinar titled Introduction to Bispecific Antibodies and Their Production. I'm Andrew Davies from the events team here at Scientist.com, and I'll be your host today. We're very fortunate to be joined by Dr. Brady Wu, head of the Protein Center at Biointron, and today he will be discussing bispecific antibody drugs and their production. Welcome, Brady.
Thank you very much, Andrew. Can you hear me clear?
We can, yes. Please take it away whenever you're ready.
Okay. It's my pleasure to be here with Scientist.com and to present Biointron for a talk about bispecific. The talk will be divided to three parts. First one is history and development. We have to go back to history to know the future. And the second part, I will introduce some of the mechanism of bispecific and also applications and why we are doing the bispecific. And the third part, I will introduce some of the uh, experience at Biointron and how we produce the bispecific for clinical applications. And also, I will take a few minutes to introduce our company. So in 2020, there is a review was a review on Nature introducing different phases of drug development. So since the development of recombinant insulin, we entered the era of biologics or large molecule, right? And they introduced a concept in this review for multi-specific drugs. That means a drug carry two different functions. For example, for ADC drug, like okay, let me draw here, like here. It's an ADC drug. It has an antibody part, which will recognize the right receptor on the cell surface. And also it contains a payload, which is often a cytotoxic drug, which will kill the cancer cell. So they define this as a multi-specific drug. And it can be a large molecule. It can also be a small molecule, like a protein. A protein drug has two parts. One part will bind to the effector, which means the degradation protein, and the other part will bind to the target here. But in a narrow sense, a bispecific, multi-specific drug means bispecific. Like here, you have let me use line here. Uh, you have a engager. One part will bind to the T cell, the other part will bind to the cancer cell. And in the narrow sense, the development of bispecific drug, like the name show here, uh, Hemibra is a bispecific drug against factor 9 and factor 10 protein that defines the age of a large molecule which contain two different parts against two different antigens. So that's the main topic I will introduce today. So let me introduce what is a bispecific antibody. First, we should know what is a monoclonal antibody. So it has two parts. The lower one is Fc part. It has a effector function. It can bind to different Fc receptors. The upper ones are Fab regions. It can bind to the antigen. So the bispecific is combined two uh, Fab part together, right? You can combine them into compose them into in different ways. The first one is called asymmetric bispecific. That means the two half of the bispecific is coming from different antibodies. And you can have symmetric bispecific. That means you add a antigen binding domain to a full antibody. And you can have fragment-based bispecific, which doesn't have Fc. So normally we'll group the bispecific into three parts, three different groups. One is called asymmetric. The second one is called symmetric, and third one is called fragment based. And to produce a bispecific, the simplest way is to express two antibodies together, right? But you will have a mispairing problem, a classic mispairing problem here. Okay, if you express light chain and heavy chain from two antibodies together, you have 16 different combinations, but only two of them are what you want. So the scientists took a long time to solve this problem, and there is a very important discovery here, uh, pioneered by the scientists of Genentech. They used a KIH and a common light chain approach to eliminate the mispairing in heavy chain, and they use common light chain to eliminate the mispairing problem at light chain. So that's a very classic format of the bispecific. And you will see again and again the similar format in our production system. And this is called the first solution for asymmetric bispecific. And this one, you add SFA fragment to a full antibody. That's called the first symmetric format. So these two are the foundations of the bispecific structures. And since this method was developed at 1996 and the patent was expired at 2017 and is free to operate, we saw a boom of different bispecific structures based on these fundamental structures. So let me go back to the biology. The bispecific was proposed by Dr. Nizonov at 1960s, very early concept. And the development of bispecific was accompanied by the discovery of TCR, because a lot of bispecific like this one, CD3, CD19 bispecific, was discovered at early 2000, and we have others like this one, GP100 CD3 bispecific, was discovered at 2012. If you go one more page into 2020s, we have CD20, CD3 bispecific, and also we have a little bit more here, like BCMA and GPRC5D as the target and the binding partner of the CD3. So CD3 is a very important component of all of the clinical active bispecific. And also we have different mechanism like this one. It's dual targeting. I will show you later on the detailed mechanism of a few bispecific as examples. But I just want to let you know the CD3 is very important as a key part of the bispecific in our real world. Okay. So based on the history, right now we have about 17 bispecific antibodies approved in total, and 9 of them has CD3 as the antigen of at least one arm. Okay, and the mechanism was very simple. You have one arm bound to the T cell, and the other arm bound to the cancer cell. So it's called obligate bind. That's why you need to put two antigen binding domain in one molecule, right? And so none of the drugs here has has one arm binding to CD3, and we have some other mechanism I will show later. For example, for other mechanisms, this one was the bispecific against PD-1 and VEGF, and the PD-1 can help the antibody bind VEGF better because the PD-1 is a receptor protein on membrane, and it has avidity effect and it can help sink the VEGF. And since VEGF is a dimer, the the VEGF, the antibody which binds to VEGF will create valency or avidity effect for PD-1. So the binding to PD-1 of the bispecific will be stronger compared to the binding to the binding of PD-1 for the equivalent monoclonal antibodies. So that's why you have avidity effect for this antibody. And recently, this antibody developed by Axo Bio has shown very good clinical efficacy. So they published a paper to explain the mechanism behind that. So this is another bispecific. It's a antibody against EGFR and c-MET. Why the authors choose to these two targets was because c-MET was a resistant receptor when you use small molecule to inhibit EGFR. So if you use a TKI to inhibit EGFR, the expression of c-MET will be upregulated, and this specific can bind to these two receptors together. Again, it's a avidity effect to enhance the efficacy of this bispecific. This emicizumab was a bispecific against factor 9 and factor 10 enzymes. When the bispecific bind to these two enzymes together, they it create adjacent mechanism to activate these two enzymes to activate factor 8 proteins. Okay, so you have to put these two antigen binding domain into one bispecific to create this obligate mechanism. So that's why we need to create a bispecific instead of two different monoclonal antibodies. So we have some newer bispecific, like effector cell engagers. I already mentioned T cell engager, and we have other innate cell engagers like macrophage or NK cells. And we have one mechanism of dual checkpoint inhibitor. You all know checkpoint inhibitor is a very successful group of biologics for cancer immunotherapy, and people tend to put two checkpoint inhibitors together, like PD-1 CTLA4 or PD-1 TIGIT. And there are more challenging combinations like co-stimulatory bispecific, like agonist, like 4-1BB and CD28. They can put them together. But they are more challenging mechanism, and they are still in clinical development. So we have all kinds of this biology, but we have to create the bispecific. As I said, there are three different groups of bispecific. One is called asymmetric here, okay? The main composition was this KIH format created by genetic scientists. And you can have all kinds of combinations. And the other group is called symmetric. That means you have a normal antibody and you add something to it. These two groups both contain the FC part, and you have fragment here, which doesn't have the FC part. The FC part is quite important for biologics because it can elongate the half-life of biologics in our body. So most of the bispecifics we are doing have the FC part. That just show the creativity of our scientists. They create all kinds of bispecific, and this page showed the asymmetric bispecific, and this page again this page showed the symmetric bispecific. But that's not quite important because as the clinical progress going on, people will find out which kind of format will be more appropriate for the biology. So that will be the all kind of structures will be focused into a few ones, which is good for our clinical efficacy. That's the most important aspect for bispecific production. So just to show you how we can produce this molecule, we have the classic mammalian antibody production process. We have heavy chain and light chain. We combine them together and transfect them to CHO cell, HEC293 cell. For monoclonal antibody, you have two chains, but for bispecific, you might have three, four, or even more than four chains. In Biointron, we made 40,000 bispecific annually, and we have produced more than 260 different formats, as shown here. And normally, we can produce bispecific products for preclinical application as fast as 2 to 3 weeks from your sequence submission. And we have a common QC for the bispecific as SDS page, SCCH, PLC, and also mass spec. For bispecific, mass spec is quite important because you often have the mispairing impurity, and it can be only identified by mass spec. So just showing some popular selections of bispecific structure. So this one was the structure as same as the drug developed by Axel Bell. It is a classic IgG SFA fusion type molecule, and it's quite popular right now. And the second one is a KIH molecule, but one arm is a VHH. That means it's coming from either llama, camel, or shark. The other part is a normal Fab. So when you combine these two together, the correct molecule will have a distinct molecular weight, and it can be separated easily by ion exchange column. And the next popular ones are this one is crossmap, and this one is a classical KIH 5 SFA fusion type of molecule, and this one is a full chain bispecific, and you, you may contain some mutations on the light chain here to avoid the mispairing. And this one is a 2-to-1 common light chain bispecific molecule. And you have more creative ones here, like a VHH fusion together and some other things. But a more complicated, the more complicated the molecule are, the more difficult you can produce them. So, for example, for IgG-SFA, we can use symmetric format, and we can fuse the SFA to the C-terminal of a heavy chain, N-terminal of heavy chain, C-terminal of the N-terminal of the light chain, and C-terminal of the light chain. And we have extended ones, like we can put KH here and VHH here, all kinds of different combinations. And also, we at Biointron, we have the llama or alpaca antibody discovery campaign, and we can produce which is a very small binding partner, and you can fuse it to a full-length IgG to create a molecule which is quite similar to IgG-SFA, but it's called IgG-VHH. And you can put in the C-terminal of the heavy chain, N-terminal of the heavy chain, light chain, N-terminal of light chain, C-terminal of light chain, so on and so forth. And you can put them in asymmetric way. And we also produce a lot of cross map, and you have different cross formats. Either you cross only CH1 domain, like this one, or you cross both domains. But we often encounter assembly problem because you cross one domain, and you may have a lot of impurity which you don't have the correct disulfide bond formed. So it's very easy to design this molecule But as I said, the more complicated the molecules are, the more difficult we can produce them. So we have a few ways to optimize the production of bispecific. The first one is quite simple. Sometimes when we optimize the ratios of the transfection of the plasmid, and it's you can eliminate a lot of impurities. And the other way is that we can use a polishing purification to get rid of the aggregates, mispairing species, and also fragments. So this part just to show two cases that we optimize the plasmid ratio to increase the purity of the first step purification. You might notice that the titer or the extraction level was not increased dramatically, but the purity was increased dramatically here, like here. So this is another case to optimize the plasmid ratio. Uh, you can notice here that for the second batch we increase the plasmid ratio of each one, which is this one. That means this one was a lower expression, which is the expression of H1 was lower than the expression of H2. So we increase the plasmid ratio and we uh, uh, had the purity increase dramatically after the first step purification. A similar case, this is a little bit complicated. It's three chains, H1, H2, and also light chain. And we increase the ratio of H2 here. That means this one is a low expressor, and the yield of after the first step purification was lowered, but the purity was increased dramatically. And sometimes when we saw bispecific antibody like this one, we can treat it like monoclonal antibody, and we can get a very high purity of just first step purification. And this one is more complicated, and we normally will analyze the SDS page and the ASCC HPLC result together. You can see it has H2Cl homodimer. That means this part was duplicated, and also the half body, which is also this part. Okay, and we have a target antibody in the middle. And we, as I said, we take an ion exchange column approach after polishing purification, and we get a high purity antibody. This one was well, quite interesting. We have this classic structure, 5 SFA-KH part, and we have a target here. We have a H2 dimer, so this part replicated, and also the half antibody of H2. So we need to remove this band, also this band, okay? And we have another cost. We have to do it with a reasonable cost. So the first approach we take is protein A as a first step purification, as always, and we use capital S as a second step. And we saw our purity was increased dramatically, but we still have a impurity here. That means the H2 homodimer was not removed completely. And we noticed that this part doesn't have a CH1 domain, like here, okay? So in our second optimization, we use CH1 as the first step, and we use capital S as the second step. And we have a quite good recovery for our purification, but the cost was a little bit high because CH1 is a affinity column, and you use a nanobody against CH1 for purification. It's not quite ideal, although it solves the problem of our customer. So we tried to optimize the plasmid ratio and to reduce the part of H2 homodimer, this band. And also we use a Hake column, a phenol Hake column for our polishing purification. and we got the end product with high purity and also good recovery. This one is a cross map, as I just said. You can see even in the non-reducing gel, the cross map has all kinds of impurities like this one. Okay. And we after the second step purification, we have the purity up to 97%, and it's good for delivery. But it's important to identify what is what. So that needs the capability of mass spec. For example, for this case of first purification, you can see the fraction of intact uh, cross map CH1 plus CL1 plus CH2 plus CL2 was only 65%. And without mass spec, we will not know do we have any mismatch product like this one. And after purification with mass spec, we will see the fraction was of the correct pairs was up to 96%, and we didn't have any mismatched pairs. Okay. And there is another way to create the bispecific. It's called controlled arm exchange. And also, often our customer will let us do this kind of experiment. It's a classical method developed by scientists of GenMab. And it's a very good method to do early screening because you can produce two parental antibodies and you just use a simple chemical reaction to create a bispecific with controlled arm exchange. So we have one client who use VHH FC for early screening, and interestingly, both of the arm needs to be in high purity, as when both of arm has very high purity, above 95%, the assembled molecule will have a purity above 95%. But if you have one below 95%, like 75%, you don't have very successful assembly. So the purity of the parental antibody is very important for this kind of approach. So that's the end of our technical discussion, and I will take a few minutes to introduce our company. Who is Biointron? We are leading CRO for high throughput antibody production and also discovery service. We are famous uh, as known that we can produce 1 week antibody production from sequence submission. We are founded in 2012. Right now we have more than 600 employees and more than 2,000 customers across 28 countries. We have a headquarters in Shanghai, China, and we have a production center at Taizhou, China, Jiangsu, and we have an office at Nanjing, and we have a logistics center at New Jersey. And we are attending a PAX meeting in Boston right now, and we have a booth there. And besides the bispecific production I introduced today, we also have other services like antigen. So we have antibody production. So we have a high throughput antibody production platform as our foundation, and we have all kind of sample generation and testing service on the foundation of high throughput antibody production, including antigen production, cell line assay development, and also discovery services like APACA VHH discovery, single B cell discovery, naive libraries, and also engineering service, including affinity harmonization, affinity mutation, and epitope binding, and also assay service like including ELISA, FACS, SPR, and cell-based assay including ADCC, ADCP, and MLR. And we are also promoting a developability service today this year. And also we have a small branch of cell line development. We have a ECACC licensed cell line, and we have a global sublicense right. So, yeah, we have expression service, monoclonal antibody expression, bispecific, VHH antibody expression, scale-up production, and cell line development and licensing, and also IDC conjugation service. For discovery, we have VHH antibody discovery, B-cell phage library, humanization, phage and also in vitro assay, biochemical and cell-based. And we have a quite capacity for binding assays, including Biaco, Katara, and also BLI. And we also have off-the-shelf products, including isotype negative control sample and in vivo ready antibody benchmark products, and also anti-mouse target antibodies. And we have a state-of-the-art production facility, including sequencer, liquid handler, shakers, and also fermenters. And we are quite automated. That's just to show you a case for our plasmid prep workstation. We have a fully automated bacterial culturing, plasmid prep, and also pipetting and labeling workstations developed by our partners, and also designed by us and developed by our partners. And we are very keen on our automation capability, because we are doing high throughput antibody production and also other services which are all in high throughput. So it's very important to build our automation capacity. With that, I will end my talk. Andrew?
All right. Thanks very much, Dr. Wu, for that fantastic presentation. And now, Brady, are you ready to answer some questions?
Of course.
Fantastic. I know you talked, there's a few good ones here. I know you talked about this a bit, but what are the mainstream methods to compose an asymmetric bispecific antibody?
So, Andrew, can you bring back the slides?
Oh, yes, of course. Sorry. Yes, sure. Here we are.
Okay, let me skip a little bit. Let me go back to the history of bispecific. So, the classic problem is the mispairing of between heavy chains and also light chains. And our binding energy was mainly contributed by the heavy chain. So since the development of the KIH format by Genentech, there are a lot of different options to create. For example, for this one, let me bring back the pointer. Okay.
Sure.
A few different options to create a heterodimeric bispecific, which are quite similar to the KH approach. But I would say the KH approach is still the most popular one. And of course, you can create your own ones as long as you didn't touch the patents of this all kinds of heavy chain engineering works by fantastic scientists. But since KH is already free to operate, and it was proved on clinical, it becomes more and more popular for neuroscientists in this field.
Okay. Fantastic. On that note, there's another question here about what are some of your best sellers or most popular items for bispecific,
I think this one
or
is for
multispecific.
yeah. I think this one is for the therapeutic drugs, right? So not our bestseller. So for this question, there are two drugs developed by Roche. This one, amicizumab, it's against factor 10 and factor 9, was sold more than 5 billion each year. Yeah, and this one also another by Roche. That's for wet AMD. This one is for hemophilia A. Sorry, let me undo this one. This one is for wet AMD. So it's an eye drug. It's against VEGF and ANG2. It was also quite popular, and the revenue number was increased dramatically. Both of the molecules were produced by Roche.
Okay, thank you very much. Are there this other question similar? Are there specific formats that are more popular, or
Yeah. In our system, yeah, I think this page showed a little bit. This one is IgG molecule, the same structure as the molecule developed by Axobio. Since the molecule by Axobio has very good clinical efficacy on uh, a very large indication in oncology, and this molecule becomes more popular in our system. So, and this one is a classic KH format, but it has a advantage for easier purification, and it has VHH and the Fab in two sides, and it's naturally eliminates the light chain mispairing issue, and it's quite popular. I would say uh, when we are seeing a good clinical results coming out for bispecific field, that kind of a molecule will become more popular in our system. Yeah.
okay, great. Are there any specific upcoming sort of developments, technological advancements, changes that you're looking forward to or expecting in the near future?
Uh, that's a very good one, Andrew. Okay, let me go back to this page. Uh, in the Nature review, it says, okay, not this one, this one. It says multispecific drugs. So ADC is one kind of multispecific drug because it has a linker payload and also a, a large molecule, right? And also bispecific as I introduced today. The combination of bispecific and ADC was quite interesting. So I'm looking forward to this field that I will say more bispecific uh, ADC molecule or even ADC molecule with different payloads.
Okay, fantastic. A question here: is your main R&D center in New Jersey or Shanghai?
It's at Shanghai.
Okay, excellent. And someone's asking about the slide deck. Just a reminder: the recording will be available afterwards. If you're interested in any specific additional details, we suggest getting in touch with Brady and the Biointron team, and they can. I'm sure they'd be happy to follow up. Maybe Brady, if you can just as we wrap up here. So if someone is interested in working with Biointron and your team, what would the process look like? They'll send you the sequence. Could you just walk through that briefly?
Yeah, if you look at our website, we should have some box you can enter the information, and after that, technical support person will contact you with all the information that we need to place order, and uh, I think all you need to provide is the sequence and also the amount of antibodies you need, and we will do the rest for you. And I think one advantage of Biointron is that uh, we even will we will even check the sequence. There's a very normal, very famous case within our system that we will uh, extract information from the patent. And you know the patent was all, a lot of patent were in the PDF format, and the printer was not very clear. And we will check the sequence, uh, the difference between two valine, and Y and one W tryptophan. So we will check the sequence for you, and uh, we have a AI system to do so, and we will have people to check on that. So you just give us your sequence and the amount you need, either volume or the milligram, and we will do the rest for you.
Okay, fantastic. So get the sequence and then you take care of the rest. I think in the interest of getting everyone to the next appointments on time here, I think we'll probably wrap up there. But I'd like to say a huge thank you, Dr. Wu, for joining us today. It's been a real pleasure having you with us, and thank you very much for your presentation.
No problem. Thank you, Andrew. My pleasure.
A big thank you also to everyone in the audience for participating. And in closing, we hope you enjoyed this Scientist.com and Biointron webinar, and we'll see you again next time.